Hello, I'm Mariah Green, and I'm a student at Northeastern Illinois University. I'm also an earth science major. I love prehistoric life, and I want to study paleontology in graduate school. I want to talk to you about the project I'm working on this summer at the Field Museum. My research focuses on Holocene fauna. The Holocene epoch ranges from 11,000 years ago through the present. The purpose of this research is to study environmental and ecological conditions of the region prior to European colonization. Weeke's Cave is a part of the Nullarbor Plain, which is located in southern Australia. We have fossils that were deposited from owl pellets, which is owl vomit. Owls swallow their prey whole, and when nature calls, they throw up any non-digestible parts of their victims, such as bones and fur. Pellets were deposited at different depths of the cave, ranging from 0 to 36 inches. We sorted fossils from rocks and sediments, so any species found could later be identified. We found many species of rodents and many species of bandicoots as well. In addition, we found fossils from birds, bats, reptiles, and even gastropod shells along the way. We cataloged the specimens in the Field Museum's vertebrate paleontology database. I learned how to describe the morphology of the specimens, whether I'm looking at the maxilla, which is the upper jaw, or dentary, the lower jaw, along with the number of teeth that the rodent or bandicoot once had. I also learned how to print many specimen labels. After the specimens have been officially cataloged, they are stored for the fossil mammal range collections forever. We plotted a principal component analysis graph for the relative abundance of the species within the depths. On this graph, we have two distinctive sets of layers, the oldest in red and the youngest in blue. These layers indicate changes in time and habitat. I also looked at taphonomy. What is taphonomy? Taphonomy is how something fossilizes over time. I analyzed the angle of breakage, perpendicular, acute, and obtuse, and the texture of breakage, smooth, step, and soft tooth, within approximately 30 femora thigh bones from each layer of the cave. Breakage pattern and texture can give us clues on how long after the animal's death was the bone broken. Potential digestive erosion within thigh bones was also examined. Results show that we mostly had complete femora and very little digestive erosion. The complete thigh bones helped us determine which owl family was responsible for the pellets. The barn owl is the owl family that likely deposited the pellets in the cave. Barn owls typically produce pellets with less digestive damage than other owl taxa. This rich data allowed us to infer more about the ecology and the environment. Who knew owl vomit could tell us so much about the past?